Father, we thank you. We thank you for the truth that you've revealed. Thank you for your word and all the riches that we have in it. Thank you that it's reliable from the very first verse in the very minutest detail. So we thank you for this opportunity to exalt you, to encourage people in the truthfulness of your word. We pray your Holy Spirit might be active in our midst, granting us ability to understand and granting me ability to communicate. So we commit this time to you. In Jesus' name, amen.
Amen.
Okay, so this is our fourth session, and I'm hoping to move more promptly today than we have the previous three sessions. I am going to do just a little bit of review for some of you who, well, for all of us. Okay. And so we've seen that in our world today there are two basic views of what the ultimate reality is. It's either God or matter. And Genesis 1:1 is a verse that speaks of this first world view. In the beginning, God created the heavens and the earth. And that's in contrast to the secular point of view today that's taught throughout our public schools, our universities, public universities, media, and it's stated here by Carl Sagan, the cosmos is all that there is or was or ever will be. So that we are basically in an effort to expose that framework as false. And basically we're focusing on neo-Darwinism, the problems with neo-Darwinism, and just as in a review, that includes Darwin's concept of natural selection combined in the 20th century with Mendel's laws of inheritance, which are true, which are valid, but added to that, Darwin didn't know where these variations, genetic variations, came from, and in the 20th century it was speculated that all these variations are a result of random genetic mutations. However, with the light of Scripture, it's easy to conclude that most of these variations we see in nature are part of the original genetic makeup of the created kinds that God created. There's in most higher plants and animals, there are 2 copies of the genome. And that makes possible a lot of diversity, built-in diversity in every type of plant and animal, including humans. So most of these variations were created in the very beginning by God, placed in the various organisms. And modern biology, modern genetics is oblivious to that. They think that these variations have risen are a result of mutation. And I'm not going to go into it again, but there's a strong case that all of these mutations that are common, that we commonly find, are part of the built-in creation from the very beginning. Okay, so many discoveries made since the 1950s now show that neo-Darwinism is bankrupt as a scientific idea, and we're considering 10 reasons why. During our first 2 sessions, we considered the first item on the list, namely, life at the molecular level is unimaginably complex. But beyond that, it is irreducibly complex. And we saw some examples of what life is like at the molecular level. It's filled with moving parts. Like this little biped robot that carries vesicles, containers containing all kinds of various chemicals all over the place inside the cell. That's going on all the time. Another process that's going on all the time is recycling of ATP, which is the energy that most cellular processes use, like those little robots we saw on the preceding slide. Each step requires a molecule of ATP, and this ATP gets recycled. The amount in our bodies every day is approximately equal to our body weight. So, and this is accomplished by these rotary machines called ATP synthase. This is what the ATP molecule looks like, and this is this rotary motor powered by protons. It's an electric motor. Recycles this ATP into a form that's usable. And in session 2, we considered how all of this machinery gets manufactured within the cell. And we saw that the specifications for each of these proteins is contained in the organism's DNA. And for a given protein, a copy is made of the relevant part of the DNA and goes outside the cell into what's called a ribosome, and the protein gets manufactured by the ribosome. So I've got a different video this week. This one has sound associated with it, and let's see. Let's see what's wrong here. Why isn't the video playing? Okay, I'm going to try to play it just by itself here. So I've got it here on my desktop. It's got the sound here. Let me just see if this will play by itself and not in the PowerPoint. Some of the Okay, this is a little animation of the ribosome, and we saw something last week.
When the RNA copy is complete, it snakes out into the outer.
All right, this is the RNA that's out of a copy from the DNA.
Display of choreography, all the components
Can you
of the
hear okay?
machine work together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles.
All right, these each have one amino acid on them,
The amino acids are the small red tips attached to the
small red tips attached to these molecules that are out in the cellular
There are different
fluid.
transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched to
These
the
each have a
RNA
three-letter code corresponding to the amino acid they're
Now
carrying.
we come to the heart of the process. Inside the ribosome,
This is the RNA going through the ribosome.
The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein
This is the
chain.
protein chain being produced.
Again, you are watching this in real time.
And this is about the rate that it happens in the
And after
cell.
a few seconds, the assembled protein
This is the
starts to
protein being
emerge from the
produced, being
ribosome.
manufactured based on the coding in that
Ribosomes use
RNA.
the same kind of protein. It just depends what genetic message you feed in on the RNA.
So if you got that,
In this case, the end product is hemoglobin.
this is
The
a
cells in our
hemoglobin in
bone
this
marrow
case.
Turn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body
There we go.
receive the
Good job.
oxygen they need.
Okay, what would you like me? It's only 2 minutes. You want me to replay that?
Oh,
Yeah,
Would
that'd
that
be
be
great.
helpful?
Yes, please.
I think you just restarted your computer.
Oh,
Oh, maybe not.
Cancel.
Undo.
Right.
I wouldn't put it in. I know.
okay. So
When the RNA copy is complete, it snakes out into the outer part of the cell.
All right,
Then,
this
in
is,
a dazzling
this
display
is
of
the
color,
RNA code
all
for the
components of a
protein.
molecular machine
Copy
work
of the
together
DNA
around
in the cell.
RNA to form
All
a
right,
miniature
this is the
factory
ribosome,
called
that-
a ribosome.
okay?
It translates the genetic information
It
in the
translates
RNA into a
this
string of
code
amino acids that will become a protein.
into protein coming out.
Special transfer molecules, the green triangles, bring each amino acid to the
Yeah,
ribosome.
these are,
The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids.
these have the code corresponding to the amino
Each
acid
transfer
built into
molecule
them.
carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape.
This is RNA pulled
The
through like a
code for
tape.
each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein
Right,
chain.
this is a growing protein chain being
Again,
produced,
you are watching this in real time.
approximately real time.
And
Speed,
after a few seconds, the assembled protein starts to emerge from the ribosome.
right here.
Ribosomes can make any kind of protein.
They can make
It
any
just depends
kind of
what
protein.
genetic message you feed in on the
Depends on
RNA.
what's on the tape.
In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Did you get that? That one of those proteins, there are 100 trillion hemoglobin molecules produced per second in your body.
Wow.
So this process you just saw is going on at just an incredible rate in your body. So that this gives you, I show this to give you a better idea of all the moving parts, all the activity that's taking place at the molecular level in our bodies to make something like as marvelous as our human body, make these be able to do what the body does. The digestion, the respiration, the vision, all the nerve activity, all of this is based on these molecular processes, these molecular machines, many of them with moving parts, as you saw here. So this is, the hemoglobin is just one of 100,000 different proteins that's in your body. So these ribosomes are able to produce all 100,000, depending on what kind of messenger RNA tape it gets fed into it. So again, so evolution says that all of this machinery somehow arose by a random trial and error process, which is utterly ridiculous. Okay, let me get back here, back into the PowerPoint.
Can I ask a question?
Yes.
This is like beyond what my brain can comprehend, but what type of degree, what type of careers require this kind of knowledge?
Well, we have, I think, a nurse in here. You would have at least a review of this. You would have exposure to all of these processes or these kinds of things in nursing. Certainly, as a medical doctor, certainly in the realm, if you're in any kind of biological research, you would need to learn all these things.
It's just amazing that we don't have a million, everybody converting immediately upon learning all this stuff. Like, it's just, I don't get it. I don't understand.
Yeah.
Man, it's in nature.
Yeah.
It's just, I know, but it's just crazy because, it's just sad.
Okay, and again, we covered this, but this is the 3-letter code that was being referred to, and this is in the RNA. So each amino acid, those little red things that were being added to the chain, is coded in corresponds to one of these three-letter sequences. There, in the case of leucine, there are 6 different letters that all words that all code for leucine. In some cases, there's only a single word that codes for one of the amino acids. But anyway, this code is crucial, and it's essentially the same code in every organism. So anyway.
I thought it was fascinating how it said there's a set of 3, right?
Right. Well, basically.
I mean, that's pretty detailed. That's just
Right.
amazing.
Well, yes, I'm gonna review a little bit about language in a moment that we covered last time. But in order to specify the 20 different amino acids, you need more than 2 letters. You have 4 different letters, and if you had a pair of them, there's 4 times 4 is 16 possible combinations. So that doesn't quite cover 20. So it turns out that God chose to use 3. So then you've got a more than you need, 64 different words available. But what this allows, and we'll have a few words about this in a moment, it allows you to have overlapping messages. Use the same letter in more than one message. You've got the flexibility to do that. But the mind power in order to produce overlapping codes is we can't even imagine that kind of intelligence. So anyway, this is the code that's used, and these 4 letters, A, C, G, and T. Somebody asked about this after class last time. Those stand for these what are called the nucleotides that form the links down the DNA chain. And the A stands for adenine, C is for cytosine, G is for guanine, and T is for thymine. And those are specific chemical units, nucleotides they're called, that form the links in the DNA ladder. So that's what these letters stand for. It's the names that were assigned to these particular chemical entities.
John, can't you make that bigger?
Let's see.
John, there is a
The
way.
presentation.
I'm sorry. Yes, I'm sorry.
At the bottom
Yes,
left.
I know it.
Yeah, there you go. No, go over further.
That'll work.
Yeah.
Yeah,
Yeah, that works. Either way.
Thank you.
I thought I had done that. I'm sorry. Thank you for pointing that out. Okay. All right. So any other questions? Are you pretty well tracking on this? Okay.
Yeah.
So we saw, in addition, I will point out, we saw just in that with the ribosome, it produces a linear chain. What begins as 4 linear chain. In the case of hemoglobin, you need 4 of those protein units to form an actual hemoglobin molecule. So what begins as 4 linear chains of amino acids folds into a 3D hemoglobin structure. The final structure depends critically on the 3D distribution of charge on the surfaces of the amino acid links forming the chains. So it starts out as a linear chain, it folds up into a 3-dimensional structure, and then 4 of these things come together to form a hemoglobin molecule. Each one has a iron atom in the middle of it, a heme unit it's called, and is able to carry an oxygen molecule. So that one hemoglobin unit like this, one hemoglobin molecule can carry 4 oxygen atoms. So it's just an unbelievable design we're seeing here. And this I showed, these are the 2 chains. There's 2 alpha chains and 2 beta chains in each hemoglobin molecule. And there's a very specific order of the amino acids. Each one of these letters corresponds to a different amino acid. And there are 141 amino acids in the alpha chain, 146 in the beta chain, and I showed you this chain here. I won't take it out of the bag, but I showed it. Who? Oh, question.
Yeah, another question. I don't understand all this, but I understand the point.
Yeah.
I think a lot of people may be in the same spot I am. Life doesn't go from disorder to high order like that. It's just the opposite, it breaks down. It
Right.
goes from order to disorder.
Right.
And if you use the example, the best example I've ever heard is if you took an airplane and took it, reduced it to all its parts on the runway, what are the chances that a storm comes through and puts it all together and it takes off? So, you know, what it takes is design. It takes a person to design that airplane and make it fly. It took something to design life.
Well, of
It
course,
didn't happen
of course,
chance.
of course. That is not, you don't need a lot of intelligence to come to that conclusion.
Right.
Bill came up with that.
Okay.
Now I
However,
want you to explain everything in that video. Mr. Brilliant.
however, be prepared to answer a rebuttal to your example. A scientist will say, but our example is organic in nature, yours is not.
Okay, you wanna hear an organic example? Take a bombardier beetle, stink bug?
Yeah, that's in Darwin's black box. He explains
Yeah.
that really well. Yeah.
Uh-huh.
So there's example, they're just saying that I've had that thrown at me.
So do you wanna explain the example?
So let me just say this chain, my wife got these at Walmart, and we needed 20, so I forget how many colors, only about 8 colors, I think. So to get more different units, we put tape around to denote the different amino acids. Anyway, this is the beta chain of human hemoglobin. And if you take the two chains together, how many possible versions of those two chains? Well, you have a total of 287 amino acids. There are 20 different possibilities at each site. You raise 20 to the 287th power, that's 10 to the 373rd power. There's absolutely nothing in the universe that could be compared with.
Is that specificity, if that's the right word, why there's no replacement for blood? My Jehovah's Witness sisters always say, you know, let me give you a card so that if I'm in an accident, you can call this number and then give replacements. I said, there is no replacement for blood. Because I've administered blood many times, if there was, we would be using it. There is nothing like hemoglobin and all that.
Hemoglobin is so complicated. The hemoglobin molecule is so unique, so that there's no way that human beings could replicate that in the laboratory. Could make an artificial version of it. Anyway, the point is that it's as I made before, and I'm not going to go through it all again. These things are so complex. There's no way a trial and error process could ever identify this. We're just trying it, just randomly trying different possibilities. There's just not enough tries. Okay. So how likely is it the random trial and error process could ever find even one biological protein? We considered how many trials are available, and we found that it can be no more than the total number of organisms that have ever lived in the history of the earth. And if we just consider bacteria, we use that number, and the total number, even if you assume 4.5 billion year age for the earth, the total number of organisms is so minutely tiny compared to the number of trials you would need to have any chance of getting even one protein, that it's impossible. Okay, so the number of trials is so pitifully small relative to the number of molecular configurations that a trial and error process is guaranteed to fail, even for one protein. And this issue alone, the lack of enough trials, is sufficient to falsify the entire neo-Darwinian framework. End of conversation. No, I would say a scientist has no rebuttal to that. There is no rebuttal. It's so obvious. Okay. This issue, the lack of enough trials is alone sufficient to falsify the, it no longer deserves status as a scientific idea. But there's another aspect of complexity of living systems that is equally devastating to the neo-Darwinian framework, and that is that these systems are irreducibly complex. Darwin acknowledged that his theory of gradual change by natural selection carried with it a stringent challenge, stating if it could be demonstrated that any complex organ, or I would say aspect of a living organism, existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Okay, so in the 1990s, Michael Behe, a biochemist at Lehigh University in Pennsylvania, wrote this book, Darwin's Black Box, and he asked the question in his introduction, what type of biological system could not be formed by numerous successive slight modifications? His answer, well, for starters, a system that is irreducibly complex. And he defines it then. By irreducibly complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes a system to effectively cease functioning. And he uses as an example the simple mouse trap. You remove any one of these parts, any one of these 5 parts shown here, and the mouse trap will not catch a mouse, regardless of how many million years you want to wait. Okay.
John, just a quick question. I had talked to Michael's colleague one time. He pronounced his last name Bay.
Behe?
No, just Bay. Michael Bay. I've not heard it pronounced the way you did. So
Okay. Well, I'm not
I
sure.
was just curious if that was
Yeah,
a misunderstanding on my
I've
part.
known him. I think he does pronounce it Behe, but
Behe, okay.
yeah, I'm not sure. I'm not positive about that. Okay, so I would say if you look at living systems, hundreds of thousands of features and subsystems within living systems are irreducibly complex. They need all their parts to work. You take one of these parts away and the entire function disappears. And one striking example is the process we just looked at of manufacturing proteins, of which the ribosome is only one component. You need the DNA, you need the RNA, you need these little what are called transfer RNA molecules for bringing the amino acids. You need the ribosome. You need you need the right cellular environment. So you take away any of those and you manufacture not a single protein. So that's an example of the kind of irreducible complexity that exists in life. And I might mention, like I did with the ATP synthase. ATP synthase motors exist in every kind of living organism, from bacteria to plants to animals to humans. And the same is true for ribosomes. That same machinery, that same, the DNA, the RNA, the ribosomes exists in every form of life. And so, if you do not have all of that all in place, life just does not occur. Life does not happen. So not only is life just inconceivably complex, on top of that, it's irreducibly complex. Okay, so I hope that registers well with you. So that was my first major point. And I've spent a lot of time on it because that by itself is sufficient. The evolutionists have no comeback, no answer to that issue. They just basically want to keep their head in the sand and ignore that problem does not exist. So in session 3, we addressed a second huge reason why Darwinism is not viable as a scientific theory, and that is life specifications are non-material and are therefore beyond the reach of any kind of material explanation like neo-Darwinism. And You know, I pointed out that most of us, we've been so conditioned in our culture to, in a mindset which says there is nothing non-material. All there is what we can see and touch and experience with our physical senses. And this philosophical framework, which says there is no non-material reality, is known as materialism or naturalism. I think the word materialism is more appropriate. So to many people, the claim that non-material reality is something we experience almost every waking moment, we participate in almost every waking moment, is a shock. You know, most people have, you know, they've never been, that claim has never crossed their comprehension before. And so I'm going to argue that language is a non-material phenomenon, and the DNA. The genetic specifications in DNA are in that category of language. They're linguistic in nature. We just looked at certain aspects. The DNA language indeed has an alphabet, which we looked at. That A, T, C, and G are the letters in the alphabet. It has a vocabulary. We looked at the dictionary of words for specifying amino acids. And it also has a grammar. And as I already said, the DNA alphabet consists of the 4 letters. The vocabulary for describing the proteins consists of 64 three-letter words. And at this point, only a few of the grammatical rules have been discovered. It's evidence that DNA has got many levels of complexity. One important thing is to know when to turn on, when to make certain proteins and when not to make them. And when an embryo is developing, going from a fertilized egg to in the case of humans, a baby that's born, there's all kinds of the process is very choreographed in detail. How you go from a single cell, that cell starts dividing, all these different organ systems start to appear, the embryo starts to take shape, the heart starts beating, the nervous system starts working. All of that involves control mechanisms, turning on and off these different proteins. All of that appears to be encoded in the DNA. So even though the protein part of the DNA represents only about 3% of the total information, it appears that most of the information involves in turning on and turning off the expression of these genes at the appropriate times. So as far as the rules for doing that, we're just starting to scratch the surface to understand how all these control mechanisms are encoded in the DNA. Okay, we've already covered this. This is actually the DNA words, which has a thymine instead of a uracil. Uracil in the RNA dictionary, the DNA has T. So I've already shown that. This is a line where instead of the letter, the words, you have the amino acids listed in the left-hand column. Okay. We discussed why this reality of there being a language aspect of life represents such a huge challenge. It has to do with the nature of language. So what is language? We covered this last time. Language involves two criteria are sufficient to define language in this basic sense that I'm doing here. The first is language involves assignment of meaning to a set of otherwise arbitrary symbols to form a vocabulary. So language involves assignment of meaning to a set of symbols to produce a vocabulary. And then a second aspect is a set of rules by which elements from the vocabulary can be joined together to create arbitrarily complex meaning structures. So I want to emphasize language is an encoding of meaning, and meaning is a non-material reality. And you know, you may have to process that a little bit. But
So
meaning,
does that mean does it have to come to a conclusion then?
Pardon?
Once you find a language and you assign a meaning, doesn't it have to be proven then?
there's no, I'm not saying, no, there's no proof of what I'm saying. I'm just making observations here.
Okay,
There's no proof. It's just observing what is.
okay.
And this is reality that I'm describing. Okay? Now examples, I used this before, examples of assigning meaning to symbols. In this case, we're talking about human language, and we're talking about spoken human language, so the symbols are sounds. Okay? And there's a certain range of meaning that for a certain fraction of the human race, you're assigning a meaning to the sound sobaka. And you know that if you speak that language, it has a very, very clear meaning. But not everybody speaks that language. In other parts of the world, the symbol would be pero. Okay, and you say that word, everybody speaks that language immediately, knows what you're referring to. And the same for all of these. In English, dog. That sound. There's nothing inherent in the dog that connects it with that sound. It's an arbitrary assignment. And in German, Hund, in French, chien, in Italian, cane, and in Chinese, gao. So, so this is an example of assigning meaning to symbols and forming a vocabulary. And then you have a set of rules, grammatical rules, to join the elements of the vocabulary together to create a more complex meaning structure. And I gave an example here, an English example. The guide dog led the blind girl through the city traffic. Now that's a fairly complex meaning structure. And you can express that with a relatively small number of words. So language is powerful, and if you have a reasonable vocabulary, there's no limit to what you can describe and express. It basically gives the human language ability gives to humans close to godlike power. And it enables all the activities that human beings are able to do. You know, I forgot to turn this on, didn't I? Or not?
What's that?
No,
What's on the screen right now?
No, I'm sorry, my sound's on. Okay. We saw that language, this general definition of language encompasses human language, both written and spoken, computer language, mathematics, and genetic language. It includes human sign language, so it's a general definition of formal language. What linguists refer to as formal language. That's what I was defining there. And it's a non-material, like I said, a non-material reality, a non-material phenomenon. Yeah, feel free to open the windows. It is getting warm. Let's see. I don't know how to change the
Hey
thermostat.
Bill, if you leave that door open and she opens the window, but there is a good cross breeze that comes through. Too loud?
Yeah, but you
Do you know
need-
anything?
Oh,
Yeah.
because you're recording.
Yeah.
I forgot.
Keep it shut if you would.
Yeah.
Never mind.
So, if you need more convincing that language expressions are something non-material, I have this illustration that a message when reliably transmitted, it doesn't depend on the carrier, the material carrier. It's identical. The message remains the same, whether it's carried by acoustic waves through the air, as is the case from my voice to your ears, or relayed electronically, which we're recording. It's going to be recorded electronically. Whether it's faxed, emailed, encrypted, sent through the mail on a DVD, or carried via smoke signals. The material carrier doesn't matter. And it demonstrates that the message, the meaning, it has a reality, but it's a non-material phenomenon. It's a non-material reality. And so, we as human beings, I would say our thoughts are in that category also, but certainly when we're speaking and writing, working on the computer, we are engaged in a non-material phenomenon. So if we as human beings are, or if we're participating in this kind of non-material reality, that's strongly suggestive that there is a non-material component to our very being, that we are not purely material entities, that we have that there's a significant non-material aspect of who we are. Of course, the Bible affirms that, indicates that we have a soul and a spirit that are non-material. So again, this is a huge problem for materialist philosophy. Einstein recognized that there's more than just the material world. He recognized that there's a logically unbridgeable gulf between the realm of concepts and propositions and the realm of what we see, hear, taste, and touch. That is the material realm. And this is a quote from he made in 1944. He says, "We have the habit of combining certain concepts and conceptual relations so definitely with certain sense experiences that we do not become conscious of the gulf, logically unbridgeable, which separates the world of sensory experiences, that is the material world, from the world of concepts and propositions, the realm, this language realm." This term Einstein gulf has been picked up by linguists and to illustrate the difference between language and the material world. So in this case, this is our dog. She was just a puppy at that point, Tiffany, and we have on the left the photo of the dog, and then on the right the word dog. A bodily thing such as a dog shown on the left exists on the side opposite the Einstein gulf from the word dog, which can be used to refer to the dog. The two entities are in separate categories of reality. The word is not a dog, and the dog is not a word. So I'm going over this again because I consider it so important, and it's something that most of us have never heard before.
Well, one is living and one is not. I mean,
yeah, but there's a more fundamental difference than that. A dead dog is still not a word. Okay? So anyway, hopefully you're getting this. Okay, now so what is the implication? Well, okay, I got another point. I included this slide we covered last time, that mathematics is language. Mathematics involves assignment of meaning to sets of symbols, specification of a set of rules or axioms for manipulating the symbols. And the point I wanted to make here is this implies that the laws of physics represent a linguistic description of the nature of the material world. In other words, the material world just like life, the material world itself has linguistic underpinnings. Those linguistic underpinnings correspond to the laws of physics. Okay? So, and I might mention here, given these insights, John 1:1 has some additional meaning. In the beginning was the Word, and the Word was with God, and the Word was God, was deity. And then you read in Genesis 1 where the world that we know, the heavens, the earth, all the plants and animals, they came into being by God saying something. And God said, "Let there be light," and there was light. And God said, I forget how many times it says "and God said" in Genesis 1, something like 10 times. Yes.
PBS had an interesting program on Nova about language and mathematics. And the issue was, are we just discovering what is real, or are we making this up? And the kind of conclusion was, we're discovering a reality here as we go along, and it gets deeper and deeper.
Right. Well, it sounds like they did not have the insight that we're just talking about. Yeah. That mathematics is language. Right. Okay. And language can describe things. And as we look at the material world, we find that mathematics can describe it. So that these laws of physics are an aspect of the world, and we discover those. We can discover those, but the descriptions, the particular symbols and mathematics we're using is something that we it's basically a human invention that we can apply here and see this correlation. So it sounds like they didn't have that clearly understood. And most people do not. Most scientists, most intellectuals do not understand these things we're talking about. Okay, we saw material processes can't generate language because it's non-material. Can random processes generate language? I showed this example. I wrote a little computer program for randomly generating sequences of English letters where each letter had its frequency taken from English written literature. And I included a space in here, so the frequency of, say, the letter H is the same you'd find in average English text, and so on. So do you see, does this as a speaker of English, do you see anything intelligible in that? No, it's just gibberish. So that's all you can expect from a random process. Random processes do not have the ability. The step of assigning meaning to symbols is missing here. That's why there's no meaning there. Okay? It's not surprising that the result is gibberish. And the reason why must this be the case? Because the defining aspect of language is missing, namely assignment of meaning. Random processes never can generate meaning-bearing strings. Moreover, and this I said this last time, random changes in text strings that carry meaning-bearing messages invariably turn those messages into gibberish. So if you start making random changes in a meaningful English text, just going in and making random changes, what's the result? Well, you turn that message into gibberish.
Does that mean we won't be able to talk to the animals?
Well, first, the animal would need to be able to, their brain would need to be able to process language. And, there is some indication that animals like dolphins, some animals do use sounds to communicate. So there is at least a very primitive level of language in animals. Our dog, can understand I don't know, 50 words, 100 words, you know, so animals have some limited ability to participate in language, but nothing like human beings. All right, so this then, if you apply this result to the genetic language and what's in the DNA, the logical expectation is if you start randomly mutating the DNA, you're going to turn it into gibberish. You should not, there's no reason to expect that that's going to lead to any kind of improvement. Okay, so I've already, I think, repeated that enough. Language involves assignment of meaning, and you need a set of rules, and that meaning is a non-material entity, and therefore language is non-material. And and the DNA specifications are linguistic and non-material, and they require a non-material explanation. So language, as we've defined it, represents a gigantic difficulty for materialist philosophy. Materialist philosophy is predicated on the belief that there are no non-material realities. Language is an irrefutable counterexample to their position, their assumption. So the claim that no material, that is to their claim that no non-material realities exist. In short, language falsifies the entire materialist framework. So this ought to be hammered home in every university, every high school, you know, anybody standing up and teaching materialism, materialist philosophy, and it's going on all around us. That whole framework is false, and it's easy to show is false. Okay? Okay, and if any of you are interested in reading more details about language, all of what I've said and more is in this paper, A Linguistic Argument for God's Existence, which was published a couple of months ago in the Journal of the Evangelical Theological Society. I'm my co-author, Jeremy Lyon. It's available on my website, logosresearchassociates.org. You have to scroll down under my name. Okay.
When we're all done with this, can you put your PowerPoint on the website
I can, yeah.
so that we could go back and reference it if we need to?
Yes, I can do that. I'll try to remember. Okay, now we're going to try to move much more rapidly. Now we're going to hopefully do 3, 4, 5, and 6 at least in the next 20 minutes. All right, the third point is namely it's the cost of substituting a new trait for an old one via natural selection. And the cost of doing that in a population is extreme. And it puts the time span required far beyond the reach of neo-Darwinism. And this is known as Haldane's dilemma. One of the fathers of modern neo-Darwinism, Haldane, realized this near the end of his career, near the end of his life. And he almost panicked because he realized his whole theory was indefensible. And that's why it's called Haldane's dilemma. Again, it's the extreme cost of replacing an existing genetic trait with a new genetic trait. And let me try to, a little more background on Haldane. He was British. He was actually a very active Marxist. He was one of the founders, 3 founders, along with Fisher and Wright, of the field known as population genetics. And his book, published in 1932, entitled The Causes of Evolution, represented a major contribution to what came to be known as the Neo-Darwinian synthesis, or Neo-Darwinism for short. It reestablished natural selection as the central mechanism of evolution by explaining it, supposedly, in terms of mathematical consequences of Mendelian genetics. Okay, so he was an important figure, in the early 20th century in establishing and ushering in this new version of Darwinism we now know called Neo-Darwinism. All right, in the 1950s, Haldane calculated the cost of fixing a new genetic trait in the population. Haldane found this cost of substitution, as it is normally called, so high that only one new trait per 300 generations on average could be selected for and become fixed in most higher vertebrate organisms. So that means that, so we're talking basically about one nucleotide, one letter in the genetic alphabet in your DNA code. Just introducing one change like that needs about 300 generations. So Haldane realized, man, evolution just can't work if it can only happen at that pace. Okay, now let's try to understand why that is. Let's try to, in our own minds, see why that's a plausible conclusion. Suppose a new beneficial trait happens to arise in a single individual. A baby's born with a mutation, or let's just call it a mutation, that gives it, say, a 10% higher IQ. All right? How does this particular trait spread to be fixed? That is, carried by every member of the human race in some future generation. So if evolution is going to work as advertised, this kind of process has to happen. But what does it require? Well, it requires that only descendants of that one individual who inherit this trait can ultimately make up the human population. Everyone else's descendants, unless they happen to marry into that line eventually, will all fail to leave any offspring. Okay? So to sort of illustrate that, suppose we have a stadium full of people, and suppose this guy in this red sweater here, suppose he's the one that's inherited this new trait that causes him to have a 10% higher IQ than everybody else. For that gene, for that new gene or that new trait to become part of the human race, means that only his offspring, only his children, grandchildren, and so on, can survive. Everybody else's children have to fail in their reproductive endeavor. They can't leave any offspring in that future generation.
He has to have incest, right?
Well, that would speed it up somewhat, but that would in general cause too many recessive mutations to rear their heads and make the resulting population not very it might more than offset that one gene. Okay. So Haldane calculated for selection to fix a new trait in the population, the number of individuals that are born must fail to reproduce because they didn't inherit that trait. That number equals about 30 times the population size, he computed. Okay? So if we're talking about a population of, say, 1 million people, for that trait to be fixed, there's got to be about 30 million individuals that can't have children that survive. Okay, so that's the kind of cost we're talking about for every trait.
John, can you apply that same theory to the argument sometimes secularists will come up with on Adam and Eve being the only two parents originally? How do you prevent recessive mutation problems, etc., starting off with only two?
Well, they didn't have mutations. They were created without genetic defects. Moreover, they were what's called heterozygous. They had different genes on the two sets of chromosomes to be able to produce all the diversity that we have today. That seems clear. And so, but mainly because they had no genetic defects, there was no problem in Adam and Eve's children marrying each other.
Okay.
Was that also because Eve came from Adam's body, so she obviously had parts of his DNA?
Yeah.
Is that true? After
But that didn't, God could, if God had chosen not to create create Eve from Adam's side, he could have created her from the dust of the ground just like he had created Adam. But for various reasons, I think there's important reasons why he chose to do it the way he did. Likely that Eve was a clone of Adam's genetics apart from the Y chromosome. She did not have the Y chromosome that Adam had. But my guess is that God used the same genetic makeup as Adam to make Eve.
Okay, is that why as time went on, mutations started to occur?
Well, I suspect there were no mutations up to the time of the fall.
Right, but after the fall, as time marched on.
And it's my suspicion that it's primarily ionizing radiation that's responsible for most of the mutations that currently exist in the human race, in the human genome.
So external effects, not necessarily internal effects.
Yes. Although I wouldn't be surprised if at the time of the fall there might well have been some genetic switches that got thrown. For example, as far as in the animal kingdom, that genetic switches that turned on traits that produce carnivory in the animal world. But it's not easy. I don't think we have the knowledge today to either confirm or refute that possibility. Yes.
Getting back to the cost of substitution, was this, how influential was this in like the genetic engineering that the Nazis tried or something like that? To try to speed things up, or was it?
They did, in general, the scientific world did not know this until Haldane applied all of his experience and realized it in I think it was 1956.
Oh, okay, so it wasn't at all.
Yeah. So the reason this 300 generations was based on, I mean, you can't take that, you don't, if you have to do away with 30 times the population size, you can't do it in one generation. And it's a heavy load if you eliminate half the population in every generation. That would mean 60 generations. It's more realistic to do it in allowing 10% of the population not being able to reproduce every generation. And at that level, you need 300 generations. Okay, but regardless of how you do it, Haldane realized it slows his evolutionary process down to a snail's pace. It makes it impossible to account for the evolution of higher animals. This probably isn't devastating for bacteria, but for any kind of higher animal, it certainly is. Okay. So this severe limit is known as Haldane's dilemma. It has been a trade secret of population genetics for the last 60 years. Basically, even with Haldane, it didn't get outside of his small community of population geneticists. Not only most scientists, but even most biologists are even today unaware of this profound difficulty with natural selection. So that's one way the secular world deals with its problems. Basically, it sort of becomes a trade secret among specialists, and that's the case here. This was not widely known. Certainly, most ordinary people today have never heard about this major problem with neo-Darwinism. So let's apply this result to the hypothesis of human evolution, that we evolved from some ape-like ancestor who lived 6 million years ago. So if we consider a human-like population with a generation time of 20 years, each generation, a new generation occurring every 20 years. Haldane's estimate implies that in 6 million years, there could have been only 1,000 DNA nucleotide replacements. If you divide 6 million by 20 and then needing 300 generations, you get 1,000. But there have been at least 75 million fixed differences. There are 75 million fixed differences at least between chimp DNA and human DNA. There's no way natural selection can account for this difference, even in the evolutionist time frame, even in 6 million years. So you can see why Haldane realized this was a fatal problem for neo-Darwinism.
Was there a reason for using the 20 years for a generation rather than 40?
No, it was just to be generous to the estimate.
You can reproduce every 20 years. No childhood divides.
So again, to repeat, natural selection has this glaring theoretical flaw: the extreme cost of nucleotide substitution makes it far too slow to work as advertised. All right, so that was number 3. Number 4, natural selection cannot even detect, much less eliminate, most bad mutations, and hence cannot prevent genetic decline. Let me just repeat that. This was something that, again, somewhat of a trade secret, and most people are not aware of it. Most biologists are not aware of this situation, that natural selection cannot even detect most bad mutations. If it can't detect them, it can't eliminate them. And hence, these bad mutations just accumulate generation after generation, eventually leading to extinction. So rather than this process of mutation and natural selection leading upward, it's constantly resulting in genetic decline, genetic deterioration, reduction in fitness. So we recall these main components of neo-Darwinism: Mendelian genetics, random genetic mutation, and natural selection. Well, a huge difficulty, just to repeat here, is natural selection cannot even see the overwhelming majority of bad mutations. Therefore, it can do nothing to halt their catastrophic effects on the genome of a population. This is a well-kept secret, even among most professional biologists, namely that natural selection cannot and does not remove the vast majority of bad mutations.
John, what do we know through observation the percentage of good versus bad mutations?
Yes, it's questionable if even one good mutation has ever been observed. So, and I'll get to that in a moment. That's another point in my 10. I'll get to that. It's doubtful that there are anything. The odds are so small for getting a good mutation by a random process. You probably wouldn't expect any good mutations. So the mutations that are commonly talked about, like resistance to pesticides, or things of that sort, if you look at what has happened in the genome, is in general some of the machinery has been broken, has been disabled, and it allows, in this particular situation, say in the presence of this pesticide, for organisms to tolerate it better. But if you put them out in the wild with that defect in their DNA, it's going to reduce their fitness. In most other situations, it involves breaking or disabling some function that's there.
So the Monsanto Roundup Ready plants, you're saying if they were planted out in the wild, they would have less chance of succeeding?
Probably.
So you're saying that's all on paper? It was never proven? about mutations.
The observation, he's saying it's questionable that they.
It's never been
observed.
Yeah, the existence of so many good mutations is just an assumption that it just, you know, the evolutionist had no other explanation for where a genetic diversity comes from. And so in the early 20th century, when it was discovered that ionizing radiation could produce genetic change, they jumped onto it. They latched onto that as their mechanism for where genetic changes occur. They were oblivious to the fact that God could have put in built-in genetic diversity from the very beginning. They were totally ignoring that possibility.
So when scientists are trying to make a rice that would grow better in India or whatever or in Bangladesh, are they taking advantage of combining more genetic code, or are they eliminating stuff? Is it becoming a narrower?
Well, what basically they're doing, they're basically discovering some genetic some versions of the, say, rice genome that God created originally that are not in the mainstream strains, the main strains that are being used today. But some of that original diversity has survived in various, you know, remote places. And as they discover that, they find under certain circumstances, certain environments, those genetic traits are beneficial. So it's primarily a matter of selective breeding and finding genetic material from various places around the earth that have some benefit under certain conditions. So that's the way that plant and animal breeders worked in Darwin's day. It's the way that's still the way most new strains are obtained today.
Don't they still find some negative things, because they're bringing in the negative traits as well?
Yeah, but with plants where you can really apply extreme selection, you can raise a million plants and just keep one. You find one. So plant breeding is you can scan for good traits and have them appear or get them folded into your strain a lot easier than with animals.
Can they do gene splicing in plants stuff too?
Yes.
So you can
Yes.
make whatever you want to make?
So yes, with the technology that now exists, you can take a gene out of one plant or even one animal and put it into another organism. So that is, and we talked briefly about how these Roundup Ready plants, how they are made. There's this gene that produces resistance to, what is it, glycophosphate? Glycophosphate. And
it's this enzyme that it affects an enzyme that's only plants have, and some plants have a different enzymatic pathway.
Yeah.
So they're resistant.
Yeah. Yes.
Would an example of what you're saying here be, to use a simple example, like when we plant tomatoes, they are generally hybrid varieties. So you plant the tomatoes, you get these big, beautiful tomatoes. You take the seeds from that tomato and replant them, and it will generally produce plants that revert back to either one of the original varieties, and usually inferior to even those.
All right. So Darwin, that's one thing Darwin discusses in his book. He was a mystery in his day. You know, the origin of what is called hybrid vigor. So if you basically, it was known back in his day. It's been exploited in the 20th century and even today of such that many crops, I don't know if most, but many crops are hybrid. Hybrid corn, for example. Almost all corn is hybrid. And so what is the origin of hybrid vigor? I would say even today a lot of biologists are unsure about that, but based on what I know the answer is relatively obvious. If you have a purebred line, basically a hybrid produced by crossing two purebred lines. So in a given purebred line, there are lots of recessive mutations. There are a lot of mutations, well, recessive, in particular, recessive mutations. Because those mutations are on both of the pairs of each chromosome, they get expressed. Those mutations come into play and reduce the vigor of the plant. Okay, but if you have two purebred lines, it turns out that many of these recessive mutations are different in these two lines. So when you cross them, you only get a single. All these recessives, or many of the recessives, are what are called heterozygous. It only occurs on one of the two chromosomes. So without so much expression of these recessive bad mutations, you get more vigor in the plant. And hybrid vigor is just that simple. It's a reduction of the expression of recessive bad mutations.
But it only works once.
But
And you replant them.
yeah, if you
and
replant
don't
them,
get the same thing.
yeah, you get things mixing together again. Okay, I'm using up. I'm not going to. All right, so we're going to. And okay. By the way, okay. I was going to advertise 2 books here, but all right. So, in the aftermath, within these small population genetic circles after Haldane, there was a real effort to try to find an answer to Haldane's dilemma. And one guy that is credited with possibly finding an answer, he didn't, but he became real famous. Japanese geneticist by the name of Kimura. In the 1960s, Kimura sought ways to use the new data pouring in from molecular biology to address issues in population genetics, using data on variation among hemoglobins in different organisms and cytochrome C in various organisms. He concluded that selection pressure could not possibly be strong enough to drive the rapid evolution the data seemed to imply. There was so much difference in these basic molecules. He concluded there's no way, especially in light of Haldane's dilemma, that you could have that much diversity. So he therefore decided that most evolution at the molecular level must be the result of random processes like mutation and drift. And he spent the rest of his life advancing this idea, which came to be known as the neutral theory of evolution. And I can't use all this time, but basically, what he showed is that that there is a box here, a no selection zone, and what we're plotting here is the frequency of a mutation and its effect on fitness. This is a 1/10% improvement in fitness. This is a 1/10% decrease in fitness. Anyway, what he showed is that for small values of effect of a mutation, there is a window here where natural selection cannot even see what's going on. And so his theory was based on a lot happening a lot of changes accumulating that natural selection couldn't see, and somehow magically
sounds like faith.
It was a desperation attempt, grasping at straws attempt to try to find some way how new mutations could suddenly rear their heads. And he's trying to do it within this no selection zone, where natural selection can't see the mutations. Okay? So basically, he can correctly be credited with identifying this zone, this no selection zone, okay, and which we're going to talk about a little more here. So experts in population genetics have been keenly aware of this reality of this no selection zone for many decades. And also that selection can't do anything about the bad mutations there. Populations geneticists have kept this important reality hidden from the rest of the professional biology community, mainly by reporting their results in terms of sophisticated mathematics in their publications.
Oh, yes.
That most biologists, that's one reason that a lot of students major in biology is there's not so much mathematics. So in most fields of biology, they're not real sophisticated in their mathematics. And so this one field that does use sophisticated mathematics is sort of beyond the comprehension of most biologists. They're not trained or equipped to understand this literature. And realizing this situation, a geneticist who had retired from Cornell, John Sanford, decided it was time for this trade secret of neo-Darwinism to be exposed so unambiguously that no biologist could possibly question the conclusion that natural selection cannot stop the steady accumulation of bad mutations in a population, and with it, it cannot stop inevitable genetic decline. And his strategy was to exploit computers and the power of computers to simulate this process at unprecedented detail. And so computer simulation provides the means to track hundreds of millions of individual mutations at once and to model the mutation selection process with incredible precision. Computer resources available today to allow us to do this with realism hardly imagined even 10 years ago. So it allows us to study the complex interaction of many biological factors simultaneously, which for an analytical solution would be cumbersome, if not impossible. Most population geneticists did their calculations just using pencil and paper. So here, Sanford said, let's use computers to model this, so we can model everything at once. We can vary parameters at will, and model this. Use the power of computers to do this much better than has been possible in the past. So we put together. He actually enlisted me to do the programming for this Mendel's Accountant. And so this is just quickly show you the results. And this is for a human-like population. And these are some of the input parameters that for this sample case we're using. Relatively small population size. We have 6, each female has 6 children. We have a mutation rate of 100 new mutations in every baby that's born on average. We use a realistic type of selection. This case is going to run for 200 generations. We use the actual human genome size, actual human number of chromosomes. We allow linkage to occur on a chromosome, chunks of DNA that don't get mixed up by crossover, what's called crossover. We even allow some beneficial mutations in this case, which is way high, at least 100 times higher than even the most optimistic estimate. We turned off recessive mutations. We use a generous, what's called fitness heritability, and we have an average negative mutation effect of like 3 parts in 10,000. So relatively mild amount of bad mutation effect. Okay, so I mentioned 100 mutations per child. Here is from the standard literature estimates of the human mutation rate. And for just the simplest kind of mutation, changing a single letter in the DNA, called a nucleotide substitution, it's estimated that there are between 100 and 300 of those mutations in every child that's born, mutations that neither of the parents had. Earlier in population genetics theory, it was considered that if the mutation rate were even 1 per offspring, it was fatal. It meant almost certain extinction of the population. But here it was shocking when they started going out and able to test it and measure it, that it was this high. Plus, there are all kinds of other type of mutations that bring that number up into the thousands. Okay, but we're going to just use we're going to use 100 in this. We just used 100 in this particular example. So what I'm going to do is show some output from this code for this example. And we're going to first look at how the average mutation count per individual changes by generation. As you have carry this through more and more generations, how does the average mutation count change with time? Well, it increases steadily, proportional to the number of generations. And so this is a graph showing how the number of mutations as a function of generation. So we go from this is first generation. This is the number of generations this way. This is the number of bad mutations on this scale per individual on average. And this is the number of favorable mutations per individual on average. So the red is the bad mutations. And after 200 generations here, the average number of bad mutations per individual is 19,802. So that means if there's 100 per generation and 200 generations, if there were not a single one eliminated, there would be 20,000. And it turns out the actual number, allowing for selection, is 19,800. So almost all of these bad mutations have been retained. They're not being selected away.
So much for natural selection.
Yeah, as far as being able to eliminate all the bad mutations. Now the good mutations, in a similar way, most of them are not being, main thing, they're not being amplified significantly. They're not being eliminated either. And so here, if you had that 1 in 10,000 mutation, you'd expect to have 2 mutations at the rate of favorables that we were assuming, and we get 1.8 in that. Okay, which makes has a negligible effect. Yes.
So I've always thought like sickle cell anemia is an inherited trait. So if it was like say 1 per 1,000, and you're seeing more cases of it, I always thought we were seeing more because there's more people in the world. So instead of 1,000 people living on your street, now there's 10,000. You're going to see a 10-fold increase. But that may not be the case. It may be because there's more genetic mutations being passed on.
Even mutations like sickle cell anemia are very rare. So it's not a result of that mutation rearing its head in new situations. Almost all of these cases are because they inherited this gene from their parents. It's not a result of that mutation occurring anew again.
It's not exponential.
Okay.
All right. So we looked at the mutation count. Let's look at how the fitness changes as a function of generation number. So this red here is a plot of the fitness as a function of generation. So we see that over this span of 200 generations, the fitness decreases about 70%. So the fitness of the individual, the ability for that population for children to be born, viable children, that fitness has decreased by 70% in just 200 generations. Now, let's consider what would happen if selection were not working at all. Mary's telling me my time's up. I'm just about here. If if there was no removal of bad mutations, the population would have gone extinct after about 30 generations. So selection is playing a major role. It's eliminating the bad mutations that are selectable that have a large effect. And so it's instead of the population going extinct at 30 generations, it's still got several hundred generations to go before you reach down in this range where the population tends to collapse and go extinct. But so let me see what I had to say here. By removing individuals with the most serious bad mutations, selection reduces the total drop in fitness to only 68% in 200 generations. So anyway, this is just to illustrate the power of this program and the general conclusions. You apply it to almost any organism, you get similar results. A famous population geneticist, James Crow, near the end of his life in 1997, published, he said, well, let's suppose we just, he was one who realized how many human mutations are occurring and was panicked almost by it. He said if you just have a 1% decrease in fitness per generation, how many generations does it take you to go extinct? And it's simple to run that calculation. It's simply 0.99 to the nth power, where ends the number of generations. And so you can see that you essentially go extinct after about 300 generations if you just have a 1% decrease in fitness. And it's estimated that something like that is taking place today. It's probably even worse than that. That on a whole, with the number of mutations that are occurring in the human genome, that the average fitness is decreasing measurably. A percent or more per generation. Now, all of this is John Sanford describes this process, which we're just looking at, as genetic entropy. Just like the information is being degraded in a genome. And he has a book published, which is fairly easy to read, called Genetic Entropy. And here you have Michelangelo's little zoom in of his painting of God and Adam. But here you see these genetic letters, A, T, C, and G, fading. So anyway, I recommend that book for somebody interested in this topic. I showed this book a couple of sessions ago by Behe, Darwin's Black Box, again. So I think that's as far as, how many generations, this raises the question, how many generations have there been since Adam? Well, if you go to the Bible, you find that there were 32 generations between Adam and David, and then depending on which line, whether it was Mary's line or Joseph's line, you either have 31 more generations or 42 generations. So, so on the order of 75 generations between Adam and Jesus. And if you have 2,000 years and 20, use a very generous number, 20 years per generation, you have probably at most another 100 generations between Christ and us. So that means there have been at most about 175 generations of human beings.
Wow. It's not a whole lot.
No. Another interesting thing, if you go to the Bible, is the kind of decrease in lifespan after the flood. In Adam's case, over 800 years. I guess this is Shem at 600 years, down to around 70 to 80 years today. Very similar to what we saw in those graphs, and John Sanford believes most of this is a result of mutation because of higher radiation levels during the flood, and also since. these, some conclusions we can reach here. It is easy to show that at current rates of genetic decline, the human race is headed for genetic catastrophe and extinction. The calculations also suggest the human race has not been around that long, probably at most less than 200 generations. Okay, so we're ready to deal next time with the, Lord willing, with this fifth point. Favorable mutations are so rare as to be non-existent relative to deleterious mutations. And I'll talk about why that is. Very easy to understand reason why this has to be the case. So I hope that's been helpful to you. And so Lord willing, we'll meet again and finish off the other 6 next week. Okay, let's close in prayer. Father, thank you that your word's true. Thank you that we can be encouraged. We can take your word at face value and be confident that your word is reliable. Thank you for our time together. Thank you for your grace and love toward us. In Jesus' name, amen.